Okay, I'm Kor-jent van Dijk. I work with Michelle at the University of Adelaide, and I will be presenting a full range population genetic study on Zostera muelleri, and it will basically be 3 studies in one, and I will go through the first 2 studies quite quickly to make it to the last bit, which is they're all interesting, but let's say the interesting bit. Zostera muelleri, covered a couple of photos here, a couple of subspecies, Zostera capricorni, Zostera mucronata, and Zostera novazelandica. And we can clearly see, like all Zosteras, we have these cross veins. And its distribution in Australia is quite broad. In the map here down the bottom, we see that it goes all the way from the Torres Strait, PNG, down to Tasmania, New Zealand, all the way to South Australia, and then there's a big gap, and you find it again in WA. It has a broad geographic range, but it's not a continuous distribution. It's generally found in estuaries and shallow bays. It's a monoecious species, and I always like to take photos of flowers, so I'll show you some photos of flowers. And here we have the male anthers, and these little fibers are the pollen grains, and these are the flowering shoots, and in the end we have these spathes that I think are the dispersal units together with several units on one of these shoots, and these are the seeds. I'll go straight into the study itself. We did a population genetics study over the whole range. As you can see, we collected 37 populations all the way down from the Torres Strait. So I started working at JCU up here, and that's when we started developing microsatellites and basically started collecting around this area. And then we moved to South Australia and started collecting around here in WA. And through, say, colleagues, we've been gathering populations from all over the range. We have developed ourselves many loci, but in this study we used 7. Craig Sherman, he developed another set of loci, and the range of the population sizes was between 7 and 53 samples, about 4,000 samples screened, 1,234 worked. And it's a fairly clonal species, and in the end we got 704 unique genetic units. Straight into the first study, and this will be the only slide I will show about this. I could go a whole 15 minutes on this, but this is the most developed of the studies, and we have a classical population genetic structure for a seagrass. So all these populations in the north are single clones, and then if we go down, we get higher clonal diversity and higher genetic diversity, and most of the genetic diversity is found around here, and then it drops down again here past the border of Queensland to New South Wales. It's strong genetic structure, but so we have these, you probably have seen these graphs. These are structure graphs, and all the colors indicate that they group together, and I've got a close-up of this, and just to understand these graphs, every line is an individual, a genetic individual. So this population is 48 samples, but it's only represented by 1 because there's only 1 genotype. The second population is the same, but once we get to Cairns, we have 8 different individuals. So if you look at these graphs, you can see more or less what the clonal diversity is by the breadth of this band. It also depends on the number of samples you collected. But you can see that the clonal diversity goes up, going south, and then declines again here. And we have a typical isolation by distance pattern, and we have relatively high genetic diversity with an expected heterozygosity of 0.5 and 9.5 alleles per locus. Then we go down to where we live now in Adelaide. Adelaide is around the orange dots. And what we see here, again, is a structure plot. And what we find is that we have basically 2 groups, populations that group together based on their allelic composition, the alleles being, let's say, the genetic characters. And one interesting fact about this is that the populations of Adelaide group together with the populations of WA. And there's a gap there, about, I don't know, 3,000 kilometers or so. It's a 3.5-hour flight. So that's interesting. I don't know if this one's related, like, they're just related. There's no directionality here, but it's something I discovered last week. So, oh, that's quite interesting. Anyway, genetic diversity is much lower. It's 0.2, so it's less than half. Allelic diversity is 3.4. And the global genetic structure is lower too. So I used 10 microsatellites. I developed a couple that Craig and et al. Developed. And what we see is that we have, for 3 of the loci, and this one's not in the analysis, but it's informative, we have a northern form, so alleles, the characters being odd in the north, for this one being odd in the north and even in the south for both. So that's something strange, and Tim Smith might agree with me, it's a pain. Figuring this out takes a lot of time because the populations don't tell it that easily. And so we got samples collected in New South Wales and some in Victoria, and we have some strange things going on over here. New South Wales has a triploid-like, tetraploid-like structure, composition. But not, so they're not the tetraploid or triploid. Some of the loci show this signal, not all loci. So I'm confident they're not tetraploids. They're behaving as diploids, but the alleles are not. I don't know. I can't say more than this. It seems, and many of the alleles in this region are shared between the northern and the southern, let's say, groups. This population structure plot is not really representative because this data, you need to use diploid data, and not all my loci are diploid. Another thing is that not all populations had the same level of, let's say, triploid, tetraploid-like combinations. Some populations were highly, let's say, multiploid, and some populations were diploid, but with a signal from both sides. If I apply this analysis on all my populations, all the way from Queensland to WA, and I force my populations to split into 2 groups, I can see that Queensland is clearly defined. And the population genetic study is traditional, it's good, no problems here. South Australia, no problems there. But in the middle, we have this intermediate signal. Again, it is not a totally fair analysis doing this with structure because it forces everything down to diploid. If I group my populations into, like forcing them into 3 clusters, you can see that the intermediate populations group kind of in the green zone, and New Zealand sits in that group too. So if you look here, New Zealand kind of groups with Queensland, but New Zealand also has this weird triploid-like, tetraploid-like signal. This is a better representation of the data. It's a PCA, and what we see here is that we have the Queensland samples, New South Wales, and South Australia. And between South Australia and Queensland, there's a really clear defined space. And what these things mean, I am not clearly, like, we don't really know yet, but they're somewhere in between. I couldn't find any, like, clear definition why this was happening. And one of the hypotheses we think might have happened is that in one of the last ice ages, Zostera muelleri went to a refuge, one maybe somewhere in Queensland, and I know this is not the real coastline of Australia in the last ice age. And we don't know if it went either to South Australia or WA. And what happened is after this ice age, it became warmer and started these Zostera populations that have been probably through a hard bottleneck, came back again and formed and reunited in New South Wales and migrated to New Zealand. That's a hypothesis. The only thing is that I can say, and this is I think the most important bit of the study, is that it's very important to know, to understand your species before you do any work. It took a long time to disentangle this. And if you're working here, there's no problem. If you're working here, there's no problem. If you're working here, it's complicated. And yeah, getting the whole picture before you start studying locally is, I would say, kind of the main message of this talk. So we've got a couple of conclusions. Queensland populations are diploid, WA populations are diploid and behave normally. Good, a couple of good papers coming out of that, I hope. But New South Wales and Victoria, I only have one population of Victoria, have this intermediate signal with triploid, tetraploid signals. There might have been an autopolyploidy event in the past, and they introgressed again and diploidized again. But we need to investigate further. But yeah, the take-home message is understanding the genetic diversity of the whole species range makes a study on local processes much more clear. Thank you. Gary, am I surprised?
No, I'm very excited by this. I'm very excited by what you're saying with Zostera because it's been picked up in Zostera marina. Royce had some data that we shared our Posidonia with him. We get some apparent triploidy occurring in Posidonia australis in Shark Bay, actually.
You see it in Thalassia, we see it in many other species. We've tried studying Halophila, and it's Halophila is very complicated, and you have diploid, tetraploid signals.
So have you actually looked at the genome size? Have you?
Not yet.
Not yet, but no. You're not expecting it to be different?
Not really, but I don't know. I'm not going to make any conclusions because I've been wrong before, so.
Okay.
Thank you. Is that it?
All right.
Kor-jent, if you did have samples from New South Wales, what would you suggest that someone might do?
Well, you can
I can talk to you.
Well, knowing the background of this, we can put them back in there. But yeah, we are developing a new set of markers using Bates, and there are a lot of nuclear single copy loci in there, and those will at least help identify at least, I guess, the number of. So there's supposed to be single copy. We have analyzed these things over the whole 1kp data set, and so they're single throughout most of the plant kingdom, and so that would be interesting to look at, and that would be a start. Maybe sequencing one. Yeah.
Yeah.
But you want to do population genetics?
If you want to do any of the omics, you've got a problem because you don't know what the lineage signal is, because you've got essentially what we think is a heat-adapted lineage, and you've got essentially a cold-adapted lineage, and they've come together in that zone. So looking at Peter's data the other day, I've got the question, wow, maybe some of this signal of up and down regulation or switching on and off could be the result of them both only present in one individual. So I would be going for using material that was either one of the other lineages first, and then comparing the really exciting hybridization design. So there's some great things that you can get out of this, but knowing that it's this zone where two shoots next to each other may have quite different evolutionary ecology history is worth
And it's so, like one population being 50 kilometers from the next can be, let's say, diploid for 8 of the 10 loci, and the next population has 8 out of the 10 being multi-peaked, and you have probably seen more than 2 peaks in your sites. And we had this workshop together to align our 2 sets of loci to get make data sets that we can combine, but we will never be able to combine them because it's difficult.
I'll talk to you later.
Thank you.